Today I'll be telling you about protein synthesis in the two parts, transcription and translation. Here I have a model of DNA, which is a double helix. Each strand is made up of three special parts: the phosphate, sugar, and the base. There are four different types of bases. Adenine, which goes with thymine, so green, which is adenine, and pink, which is thymine, and cytosine and guanine, which blue is cytosine and red is guanine. Okay. Each, when put together, form a weak hydrogen bond. Next step, let's go into transcription. So what will happen to the DNA molecules? It will be split down the middle, unzip. And then we'll move into transcription. The two differences in these strands is that this strand of mRNA is made up of ribose, while this is deoxyribose. And instead of the base being thymine, is now uracil. Once each is completely paired with each other all the way down the line, the mRNA then moves to the ribosome to begin the protein synthesis and translation. Now we begin the process of translation from mRNA to amino acid. This is the ribosome, where the process for translation will occur. It begins when three nucleotides of the mRNA strand enters the ribosome. They put three nucleotides, and together they code for a corresponding amino acid. The first nucleotide, AUG, is the start codon. That means that the process of translation can occur. This is the tRNA, which contains the anticodon for the codon of RNA. It also contains one amino acid, which is different for each codon. Once it is bonded, the ribosome can move on down the line to the next codon. Another tRNA will join all the way down the line until the last strand. And as this occurs, dehydration synthesis will happen, and a peptide bond will form between these two amino acids. The first tRNA will leave, and the ribosome will continue on. The last codon is called the stop codon. It does not have an amino acid. Instead, the process of translation ends, and the amino acid chain breaks and forms a complete polypeptide chain or protein.